introduction. So I have no disclosures. The only thing, as I mentioned, the only disclosure I have is, as recently mentioned, I moved to the University of Utah from Alberta this past fall. And many of the pictures, including my title slide, are from my former neighbor, who's a professional photographer up there in Alberta. So I like showing his, but I thought I would be amiss not to show my new home. And so this is the University of Utah here up in the northeast quadrant of Salt Lake City. This is Primary Children's Hospital here. And where our clinical department is, and the Huntsman Cancer Institute sort of backs up onto the mountain there. And so from my lab window, I can look out onto the Bonneville Shoreline Trail, which is always calling me for a hike. It's a little hard to stay at work some days. But. All right, so as was mentioned, so my background, and maybe this is more for the trainees, so sometimes you never know what path your career will go. So after doing pediatric pathology, I did sort of a mini fellowship in bone pathology at Mayo Clinic with Dr. Uni, and then got into the molecular genetics of skeletal anomalies and cartilage tumor syndromes, particularly during my postdoc with Matt Warman at Harvard. And after that, in making some different discoveries, I started working in vascular anomalies just because there was a clinical need for someone to help pick up the volume. And so I got started on that. And then lo and behold, someone said, hey, can you combine those fancy genetics with vascular anomalies to help these patients? And so that led us to work on these low-level somatic mosaic conditions and do that. In the past few years, I've sort of branched out even more, again, by someone just asking for some help and some friendly collaboration on working for different neurocrystopathy-based disorders, which I won't be talking about today. And we're doing a lot of sort of single-cell resolution stuff in that right now. So today's talk is going to be more of a bit of a broad overview because I know this is a mixed audience. So it's not going to be a lot of histopathology or molecular genetics and things. It's going to more tell a story about discovery within this field. So we're going to start by looking at some embryology, reviewing the embryologic origins of somatic mosaic conditions, focusing on the vascular anomaly overgrowth disorders that I've been studying. We're going to review some relevant clinical and pathologic features of these disorders. And then I hope what you understand by the end of this is that in addition to being benign conditions, quote unquote, with significant morbidity and even mortality, that these are cancer predisposition syndromes. So we'll be looking at the clinical in that with the note that some of these phenotypes are a bit severe. Okay. So let's dive into mosaicism now. Now, when we talk about mosaicism, I want to sort of spell it out. There's various different types. There's type 1, there's 3 different types of type 1 mosaicism, and type 2 mosaicism, if you want to go back to sort of your classical genetics learning and understanding. And the one probably many in here at an adult hospital think about is type 2 somatic mosaicism. This is like cancer predisposition, classical syndromes, wherein we have an embryo here, and let's say this embryo is highlighted pink, because this is a heterozygous embryo, so it carries either an inherited or a de novo variant, so there's a mutation in here. Typically, we think of this as a tumor suppressor loss. So it has one hit in every cell in the body. So the embryo grows and develops, and either prenatally or postnatally, in a single cell or in a region of the body, you can develop a second hit. That second hit may be in the same exact gene or another gene in that pathway to knock it out, thereby developing additional tumors, right? So that's classically what we think about in cancer predisposition syndromes. And the one I want to illustrate that today for purposes is PTEN. So the PTEN Hamartoma Tumor Syndrome, which I'll just mention as PTEN now, has been well characterized for many years. And I want to show some salient features of PTEN now. So this is actually Ms. Cowden from Lloyd and Dennis's 1963 publication, where she was described in great detail, sort of, completely nude there on the front page of the article, and having macrocephaly, and she was described as intellectually dull and uninteresting, which we know now that PTEN patients have often autism spectrum and some degree of developmental delay. They often have all of these adenohamartomatous lesions, for example, cutaneous papillomas showing here, trichomonas in the skin, penile and vulvar lentigines, For the neuropathologist, there's dysplastic overgrowth of the cerebellum, so Lemay-Duclos disease, as well as all of these visceral adenohamartomatous lesions in the GI tract, thyroid, uterus, breast, and soft tissue. Now, these are all the result of a first hit that the patient is born with, and then second hits will then confer malignancy at varying rates, ranging from about 5% in the skin to about 80+% in the breast, right? So a very high incidence of developing certain carcinomas in adulthood. Interestingly, though, for the stuff we're talking about today, these patients do not develop vascular anomalies or tumors. So you don't see liposarcomas and angiosarcomas in these patients for some reason. All right. So a little over a decade ago now, I worked initially with the vascular anomalies group in Boston when I was there in interventional radiology, the clinical group, and then eventually pathology. We characterized this entity described as the PTEN hamartoma of soft tissue, which I'll otherwise call as FOST now. This is an early childhood lesion that often precedes other manifestations of PTEN. So if this is not a known PTEN family and they have this weird overgrowth, I'm showing extreme examples here, but they can be relatively small. And so they're important to identify. They typically occur in the extremities, either upper or lower extremities, although we can see them in the trunk. And these lesions are quite infiltrative. So if you look here, this is a patient who was severely affected in his lower limb, went on to have an amputation, and look at the skin, the subcutaneous tissue, the adipose tissue, and even intraosseous compartments are completely involved by this malformation. So this is a quote-unquote benign condition, but extremely infiltrative and locally aggressive. I'll review the histopathology on the next slide, but what I want to show for FOST is it has a very rather characteristic imaging. This is an arteriogram where you can see the dye injected here, and we have this very tortuous artery winding around and suddenly draining into this abrupt compartment. Now, a typically AVM will shunt right through an arterial component, through shunting vessels into a venous phase. This PTEN hamartoma has a much more direct shunting that's quite characteristic on imaging. So often I rely on radiologists to help with that diagnosis. So what does it look like? And so here's some of my histopathology for the talk. This is actually a low-power scanning micrograph of muscle, believe it or not, completely replaced by fat and fibrous tissue, as well as a vascular component. And what does that look like? Well, these are very thick-walled, tortuous arteries, so those things we saw in the arteriogram. The smooth muscle actually onion skins out and sort of radiates out into this myxoid soft tissue around them. Additionally with that are these dilated vascular lakes, which is debated whether or not these are venous, lymphatic, or hybrid type channels, although we think our best understanding is that these are venous pools that just sort of accept this blood. There are also some angulated, thick-walled, and indeterminate shunting vessels as evidence of the shunting phenomenon that goes on within these. But then the hamartomatous features can often be quite prominent. So this is like a neovascular genesis and angiogenesis happening. So you have this primitive myxoid matrix and little vascular sprouts that happen. This is a common phenomenon in these lesions. This is perineurial proliferation, looking like a perineurioma almost. You can see metaplastic bone and cartilage. Lymphoid aggregates, even germinal centers are common within these. So they have this weird shunting as well as a hamartomatous component to them that's quite distinct from a classic AVM. Now, I want to spend a little bit of time here just on the pathology before we go on to other types of mosaicism. So back how we defined FOST is my mentor, Harry Kosakevich in Boston, had these boxes and boxes of unusual AVM-like lesions and funny vascular things. And I went even as a visiting resident and then spent time there. And we would sit through and go through all of these and try and parse out which ones fit together. And so we came up with this group that all looked like FOST. We were blinded to the clinical information. And then when we went back and looked at that, we found that the majority of patients indeed had PTEN. But there was another group that had lesions that looked very similar that had no stigmata of PTEN Hamartoma Tumor Syndrome. They were otherwise asymptomatic with a solitary mass. And this entity is what became known as FAVA, or fibroadipose vascular anomaly. I apologize for all of the acronyms in this. None of them are mine, but hopefully the legumes will help. So FAVA, like PTEN, typically involves the extremities. We can see here in the upper extremity and lower extremity here. And like PTEN, it's an infiltrative lesion. You can see the gastroc here is completely replaced on this MR. You can see the high-intensity fat signaling and fibrosis, as well as some flow voids from the vascular channels. If you look at an imaging here, again, you can see this tortuous vessel draining into this dilated component. However, this is a venous phase image. So these are low-flow, venous-based malformations, not arterial venous shunting like we see in FOST. And that is the most key differential I usually offer people to distinguish between FOST and FAVA, is by flow characteristics can be very helpful. Here, this is actually this patient's gastroc when they went in for resection. So they can try sclerotherapy, cryotherapy, and other interventions for these, but often since they're so infiltrative, they lead to incredibly painful contractures and many times have to be surgically resected. I actually have a gastroc like this on my gross bench last week for a FAVA, so it's not that uncommon specimen at a children's hospital. All right, so just in comparing the two entities again, FOST is syndromic. It can be painful, can be multiple, and it has this infiltrative growth with an AV shunting pattern, whereas FAVA is non-syndromic, often exquisitely painful, usually singular, intramuscular, and it's a more venous phase. And the histology of FAVA looks a lot like PTEN, as I mentioned. It has these dilated venous pools here, very thick-walled vessels like we see in a FOST, as you can see here. But if you do an elastic stain, which I'm showing in this image, you'll see there's no elastic lamina here. These are arterialized veins, and so it is a venous process. There's also a venolymphatic component to FAVA that we're now appreciating much more. And another way to distinguish them now is with molecular. So a number of years ago, we showed that many FAVAs are associated with PIK3CA hotspot mutations, whereas FOST is obviously due to PTEN. All right, so a little bit then about FOST, PTEN in the setting of type 2 mosaicism. These are some of the other lakes, the Vermilion Lakes in Banff, looking onto Mount Rundle there. All right, so now let's talk about type 1 mosaicism, starting with the most severe form, which would be disseminated mosaicism. So let's go back to our little cartoon diagram here and picture we have our embryo, except this embryo does not have a constitutive mutation. It's otherwise quote unquote normal, let's say, and somewhere early in development acquires a hit. Wherever that cell's progeny are within the developing embryo will carry that mutation, right? Typically, we think of these as oncogenic activations rather than tumor suppressor losses in this, with the thought that if this occurred constitutively, meaning every cell in the developing embryo had this mutation, it would be embryonic lethal in most conditions. You have to sort of dial down the dose of mosaicism for this oncogene to allow survival, although you will definitely have a phenotype that's determined by the pattern of mosaicism. So that's disseminated mosaicism happening early in development. And an example of this, which we've studied over the years, is known as CLOVES. Again, another acronym standing for congenital lipomatous overgrowth with vascular, epidermal, and skeletal anomalies. CLOVES, the acronym, and the syndrome was coined in 2008, 2009 by a series of publications, although looking back in the old literature, there's actually exquisite publications of CLOVES patients from the 1850s in the German literature, for example, with really nice detail. So here's some features of CLOVES. You can see this is disseminated mosaicism, quite a significant and severe phenotype in many individuals. This congenital lipomatous overgrowth is asymmetric, often involving the trunk, again, just due to the pattern of mosaicism. One thing we didn't appreciate early on in the descriptions of CLOVES was that their heads were also big. It was just hard to see because of all the other overgrowth. So hemimegalencephaly is also a common finding, as well as many different acral anomalies. You can see macrodactyly, syndactyly, polydactyly, what the geneticists will call a sandal gap deformity, or a widened space between the first and second toe. And then all of the vascular anomalies associated with this. So cutaneous capillary, lymphatic stains. This is an epidermal nevus. Just look at the difference between the normal and the overgrown foot in this individual. Now, because this is the result of disseminated mosaicism, this young child here will never develop overgrowth of her upper extremities because the pattern is set during development and they are normal, meaning the mutation shouldn't be in there. But as she grows into adolescence, will look a lot like the individual on the right side because this is progressive overgrowth over time. Now, what gets these patients into a lot of trouble and even kills them is what you don't see. These overgrowths will actually grow down in across tissue planes about the spine, causing severe kyphosis, scoliosis. They often have significant vascular anomalies about their spine and pelvis, Look at this massive innominate vein here and axillary venous system on this patient. So thrombosis is a common means of mortality in a closed patient. And obviously these patients are taken to the OR very carefully and sparingly and only if needed. Now, as a pathologist, looking at the histopathology of these is actually quite bland and not very exciting looking. On the upper side here, this is the skin, a relatively banal dilated capillary malformation. But if you look down deeper, what you start to notice is there's too much fat in here. The fibrous tissue is definitely displaced. And if we look here, this is a lymphatic channel, a venous channel. There were these capillary channels, fat and fibrosis. So whenever you see a combined malformation like this, even though it looks totally bland, a garden variety venous or lymphatic malformation shouldn't look like this. So when you see something like this, you have to start thinking about one of these anomalies. All right. So that was disseminated mosaicism in CLOVES. We've got 2 more to cover. And now segmental mosaicism is another type of type 1 mosaicism. Now let's say instead of that mutation occurring early in the embryo, where it occurred later, like in a developing limb bud, so that only that limb in the body is affected and the rest is normal. So an example of that, which we've studied over the years, is Klippel-Trenaunay syndrome, or KTS. And this was described in the French literature by Klippel and Trenaunay in 1900. In well-characterized series. This is just a series of KTS patients from the Boston group when I was there. And classically, what we think of Klippel-Trenaunay is the image here in the upper left, where there's overgrowth just of one limb, as you can see here. And there's also this lateral marginal venous anomaly that goes along with this. And this is due to a developmental anomaly of the lateral marginal vein of Seville, for those who are interested in that. And it can wrap up around the pelvis. The overgrowth can be quite massive in KTS, and it can even be bilateral. Such that to the point when we were first trying to parse out these things in our vascular anomalies clinic, we'd say, all right, this patient has KTS on the left, this patient on the right clearly has CLOVES, and you meet somewhere in the middle and try and decide, is this CLOVES or KTS? It became a bit of a moot point later, once we understood the genetics of these, but in my simplistic way of thinking about this, I like to think of KTS as CLOVES from the waist down. You shouldn't see truncal overgrowth and other things up above. It's clearly something that happens in the lower extremities and limb buds and not in the pelvis and abdomen. All right, and finally, a point mutation. And this is, again, what we think about as pathologists for a lot of different types of cancer, where you have an oncogenic event, either later in development or postnatally, causing a mass-type lesion. And the one I want to bring up for this as an example, again, what we've studied in the lab are lymphatic malformations, right? And so lymphatic malformations are benign, certainly proliferations or disorganizations of lymphatic channels, but as you can see from these examples, have significant morbidity associated with them. We typically see them in the head and neck, where they can be quite morbid, especially when involving the tongue and airway. Another common place we see these is in the mesentery. So I always like to tell my pathology residents, if you get a specimen that says mesenteric cyst, write lymphatic malformation in your diagnostic line before you even look at the slide, right? Because there is no such thing as a mesenteric cyst, right? It's always a lymphatic malformation. All right, so that was 3 different examples of type 1 mosaicism. So disseminated, like CLOVES, happening very early; segmental, like Klippel-Trenaunay, happening maybe in a limb; and a point mutation, like such that occurs within lymphatic malformation. Now, about a dozen years or so now ago, we, in studying all of those different conditions, found that they were all associated with a particular subset of activating PIK3CA hotspot mutations. And remember, PIK3CA is the catalytic component of the PI3 kinase complex that sits at the base of receptors that binds things like EGF, VEGF, angiopoietin, activate AKT-mTOR signaling, and regulate cell size, cell growth, proliferation, angiogenesis, vasculogenesis. So not surprising that aberrant activation of this pathway would lead to overgrowth and vascular anomalies. And then a little while after, the series of publications that our group had put out, as well as some groups in the UK, LA, around, discovered that many different overgrowth vascular anomaly conditions were associated with activation of PA3 kinase AKT-mTOR signaling, either directly due to PIK3CA or due to AKT, or even loss of function of PTEN. And a consensus conference was held at the NIH, and it was determined that to put these under the rubric of the PIK3CA-Related Overgrowth Spectrum, or PROS. Because clinically, they should all be thought about as sort of a spectrum, because they're all related to the underlying mutation. Just a few other examples here of some shots from Alberta, just to break it up. So despite the bow tie, I am a hockey player. although this is my neighbor again. And the nice thing on the lakes up there is often they freeze so early because it's so cold, and they freeze before the snow. And many of the lakes up there have these methane gas bubbles, and they'll freeze as well. And so you can be out on the lakes skating on all of these different bubbles, which is really cool thing to do. Highly recommended, as long as it's warm enough. All right. And then what about when we found these mutations, what was the allele frequency behind them? If you look at a CLOVES patient and look at tissue between that, you may find like 15% of the cells in that tissue carry that PIK3CA mutation. It might be something like 5 to 10% for a KTS patient, and usually for a lymphatic malformation, we're talking about just a few percent of cells carry that mutation. Now, I'm not going to spend too much time on molecular detection of these things, because I think many of you are probably well versed in molecular techniques now. But back in the day, when we were first looking at these things, to understand and detect these and validate our things, you had to have a sensitive method to do this. And remember, our classical way of validating things would be by something like Sanger sequencing, right, which you can detect any mutation by, where you take your genomic DNA, PCR up millions of copies of several hundred bases that flank your mutation, run a dideoxy sequencing reaction, and pass it through a capillary electrophoresis system with a laser detection. So that, for example, if this fragment here, 50% of the fragments were green and 50% were red, the laser would be able to pick that up and you'd see it and be able to detect that mutation. But if 1% were red and 99% green, you'd never be able to detect that. And so back in about 2010, we started working with Bio-Rad on droplet digital PCR as a means for low-level somatic mosaic detection. And imagine the way I like to explain it is this, is that if you had that same tube again where you had one mutant fragment and 99 regular fragments, again, you can't detect that if you try and analyze all 100 at the same time. But what if you parsed each piece of DNA into a little droplet or well, and then ran a reaction like a hydrolysis probes or TACMAN type assay that can distinguish wild type versus mutant with different fluorochromes, and then asked, is this well wild type or mutant? Is that well wild type or mutant? Then you're reporting out at a single fragment resolution. So it's like a massively parallel-based system for detecting this. And then you can plot out wild type versus mutant and easily be able to see this. And this is quite sensitive. So here's an example of a low frequency positive, about 3%, where you can see about 10,000 copies of the DNA in this sample were wild type and about 300 mutant. This is a much lower at about 0.5%, where 12,000 are wild type and only in less than 30 are mutant. So very, very sensitive for detecting these very low-level mutations. And obviously, as Jimmy in the group here and Jimmy Bennett in the group here in Seattle has shown, this is just one of their figures, you can now take this, multiplex this, and do this clinically in a cell-free DNA-based system. Back when we first started doing this, the cell-free DNA technology wasn't quite as good, and we tried looking at urine and other means to detect these. But now, luckily, you can sometimes spare these patients from a biopsy to be able to detect these mutations. All right, so just a little diversion there on understanding a little bit about the genetics of those. Some northern lights, of course. All right, I want to talk about pathogenesis and mosaicism. A lot of this is a bit of hand-waving here. There's some preliminary data out there, but this is an area where after all this discovery was done, there's a lot of work that's ongoing and still needs to be done to figure out what's going on to cause these overgrowth and vascular malformations. So you remember in my initial description of mosaicism, if you have what we thought naively maybe in looking at this was that, okay, if you biopsy an overgrown limb in this patient, you're going to see the mutation maybe in most of the cells in that, right, because it's very severely affected. So we thought, okay, almost all the mesenchymal cells in that limb will be affected. So maybe in a cartoon format with the red cells being affected would look something like that with our cell population. So we did some initial painstaking macro dissections, later went on to sort of laser capture micro dissections. This is a CLOVES patient who had a below-the-knee amputation. This was a massive specimen. You can see the tibia, maybe make out the fibula there. It's kind of hard. And just notice this massive overgrowth and dilated vascular channels here. We dissected out the skin, the capillary malformation, the large vessels, the joint spaces, the synovium was involved, the cartilage, the fat, and every compartment found the mutation no matter how much we looked. So clearly it wasn't just the endothelial cells or the fat cells that were carrying this PIK3CA mutation. It was all types of cells seemed to have it at varying degrees of levels and lower than we thought, so maybe something like this. But then remember I said that for something like a lymphatic malformation, it can only be 1 or 2% of the cells. So in reality, what's going on is something like this, where you just have a few mutant cells impacting neighbors, which are all wild type, and somehow causing these massive overgrowths. So why are there so few cells if you have such a severe phenotype, right? You know, and initially we were trying to think, oh, is there some kind of negative selection, genetic rescue, stem cells coming in, maybe overpopulating wild type? Was our hypothesis of mosaicism completely wrong? And then how are there so few cells? I mean, look at this phenotype. How can that be caused by 1 or 2% of cells, right? And then, you know, the thought paradigm sort of shifted that probably this is some kind of early programming and patterning that once it's set in place, there's what's called non-cell autonomous effects on the neighboring wild type cells, and this can't be changed because now the microenvironment surrounding these cells is now abnormal. Now we have a little bit of data on this, mostly just from PTEN, and this was a collaboration I did with Carol Shermans, who's at the University of Toronto, and she's a retinal biologist, and we made one of the first, the first animal model of a PTEN hamartoma, but we did it in the retina just because that's what her lab was set up for. And this is just a squash mount of the retina, and by knocking out PTEN in the peripheral retina, a hamartoma actually forms in the central retina. I want to show you these images a bit better. So this M cherry signal, the red, is the double PTEN knockout in the peripheral retina, and the central retina here is where the hamartoma is forming. But if you look at that, most of the cells are green with just a single hit of PTEN, and it's only a few double hit cells within that that cause the hamartoma. So clearly there's an abnormal microenvironment where these quote-unquote wild type, or in PTEN case, single hit cells are being recruited to form a hamartoma, even though they should otherwise be behaving normally. We had some really nice preliminary data to explain this. Unfortunately, CIHR didn't really want to fund it, and we sort of dropped it a bit. But what I think is going on, and what we at least had some data to support, is this. In that, remember, cells are secreting and picking up things all the time, right? And within cell biology, we talk a lot about microvesicles and exosomes. So these are lipid membrane-bound things that are exocytosed out of the cells, depending on the type, which pathway it goes on. And they can put in DNA, RNA, and protein and sort of ship this off to neighboring cells and be taken up. And we had really nice evidence to show that the PTEN double mutant cells were actually packaging things and shipping them to their neighboring cells and hijacking their cellular machinery to making them look like they were mutant cells. So probably something like this is going on within these overgrowths, although I think a lot of work needs to be done to further explore these mechanisms. Now, what other examples do we have of non-cell autonomous effects within vascular anomalies and overgrowth? So a nice example of this is what occurs in venous malformations. So in a venous malformation, we have these very dilated venous channels. Now, typically a vein has lots of nice, smooth muscle, pericytes compacted around it to keep it nice and taut. But in a venous malformation, they can get very large and cavernous in appearance. And if you do a smooth muscle actin stain, for example, for pericytes, you'll see there's a lot of non-compaction of the muscle, so they don't form a normal cell wall. And the reason why they're doing this is because the mutation in the endothelial cells and a few cells surrounding that are creating abnormal gradients of PDGF and other things that fail to recruit in wild-type cells to form that nice coat. So thereby, again, a few bad actors are sort of changing the whole microenvironment. One other example of these non-cell autonomous effects, and this is some from the earlier work I did with Judith Beauvais and Leiden on cartilage tumors, is we showed that when cartilage tumor syndromes have a high rate of developing secondary chondrosarcomas, but if you look at the molecular genetics of the chondrosarcomas, many of those are actually wild-type cells that have transformed into the chondrosarcoma, not the mutant, benign mutant cells going that way. And that's just due to, again, an abnormal microenvironment within the cartilage matrix causing that. So just some different examples of these types of interactions. Again, I think a lot of talented work is being done on that now, and I think a lot more needs to be done to figure out the mechanisms. So just some of the many scenic drives up there in Alberta. I was lucky enough last summer to take my 2 then 9-year-olds and hiking up to the Taylor Glacier here. We got to do quite a lot of glacier hikes when we were up there. They were pretty good at a 10, 12-hour day ordeal. But many may recognize behind these mountains, if you keep driving on the road without any hiking involved, you can get to some spectacular lakes like this is Moraine Lake, which is a sister lake to Lake Louise, with this beautiful glacial silt in the water there. You have to get there very early to find a parking space. All right, so I want to talk now to sort of try and put this stuff together in understanding malignancy versus malformation and the paradigm of that. So when we first found these PIK3CA variants, this was quite a while ago, and we actually thought we had sequence contamination, and we sat on it for 1.5 years because the Harvard facility at that time had a lot of trouble with contamination, and we were like, no way, this is not real. This is not what's causing this. Why? Because those same 5 or 6 PIK3A hotspot variants, and many may know in the room better than me, are behind 50% of endometrial cancers, 40% of cervical cancers, a driver mutation from pre-invasive to invasive, a small percentage of very aggressive non-small cell lung cancers, 25% of colorectal cancers, a third of breast cancers, 90% of malignant brain tumors have these exact same mutations. And we thought, oh, there's contamination because someone else was doing a colon cancer project on the same run as ours. And so we said, oh no, this is what. Then we kept doing it and repeating and finally believed it. And then we said, okay, if that's true, then it must mean that patients with PROS have an increased risk of developing breast tumors, endometrial tumors, right? Because they have these PIK3CA mutations. They don't have a higher incidence of developing any of these malignancies, right? It's not at all above baseline from what we can see. What they develop instead is Wilms tumor, which was rather interesting because PIK3CA is not one of those genes you think of when you think about Wilms tumor. So it was known from the beginning that renal asymmetry and involvement was common in PROS, which I sort of glanced over at the beginning. You can see here a kidney and then a hypoplastic kidney on this side. You can see renal cysts involved here and multifocal renal masses. So nephroblastoma and Wilms tumor is seen in maybe about 5% of patients, especially with something like CLOVES. This is just a small published series, and I looked at the pathology of most of these cases. And what I want to highlight is just this. If you look at the age of onset here, 1 to 2 years of age, classically we think of Wilms tumor as happening in kids usually 3 to 5 years of age. So this is earlier than normal. And then if you look at the hypertrophied kidney versus the tumor location, it doesn't always match. So the classical paradigm in overgrowth is that kids with overgrowth in Wilms tumor develop Wilms tumor in the overgrown kidney, right? Because it's hyperplastic, it's proliferating. In PROS, that's not the case. The histopathology of these for the pediatric pathologists look relatively banal. They're quite favorable in appearance. So over the last years now, it's seen that CLOVES definitely has a risk of maybe about 5% of Wilms tumor. Other conditions which I haven't talked about today, which have some degree of mosaicism, also have a risk, whereas something like Klippel-Trenaunay and FAVA do not have a risk of developing Wilms tumor. And if we think about it, if you have disseminated mosaicism, it's more likely to involve the kidney, right? So chances are you develop a Wilms tumor. But if you've got a malformation that only involves your lower extremities, probably you have no mutation in your kidneys, right? So you won't develop Wilms tumor. So that's just a simplistic and likely, explanation for this. Now, how does this compare to classical Wilms tumor syndromes? So the one we think about most on pathology boards would be something like Beckwith-Wiedemann. Higher, probably double the risk, maybe about 10% risk. Early onset like PROS patients, but typically the Wilms, and almost always the Wilms tumor is occurring in the hyperplastic kidney, right? There are other disorders, maybe a bit of a wastebasket where these things are mentioned, which also have overgrowth and Wilms tumor. I think more needs to be done to figure out what's going on in those. Right, so we see Wilms tumor in PROS patients and not all of those other things. Why is it we see Wilms tumor and what's so special about the kidney in developing in this? And this is where I'm again going to go off on my sort of hand-waving pretty pictures to say that, we don't really know what's going on, okay? But it's a rather interesting thing to think about. This is what we do know about Wilms tumor. And again, this is not my area of expertise. But if you remember back to your histology days, we have the Wolffian duct here and the ureteric bud, which is going to form the kidney. The ureteric bud is surrounded by this primitive mesenchymal or blastemal type cells, which then differentiate into primitive metanephric mesenchyme. These then undergo epithelial to mesenchymal transition and form tubules in a process that takes about 36 weeks of development. So by the time a child is born, most of their everything has formed tubules, right? Now, if you look at the essential genes in this, you won't see PIK3CA. WT1 is a gene we certainly associate with that. Wnt beta-catenin signaling is important in that, as well as other embryologic genes like the 6 genes, growth factors, histone modifiers are all critical in this stage. Now, Wilms tumor is one of those classical pediatric tumors that's called an embryonal tumor, right? These are like tumors that are tissues that never stop developing. And what do I mean by that? Well, a Wilms tumor, if we think about it, forms because some point during development, you're maintaining expression of these primitive genes that promote this stem-like phenotype, and these keep proliferating and developing a tumor, or you're somehow blocking expression of the genes. You're failing to either turn those off or blocking expression of new genes, which will induce this tubular differentiation. And then you'll be stuck in a phase with the tumor development, or you actually can get re-expression of some of those early genes at some point in development. Now, the key here with these embryonal tumors is that turning back on, if that's the phenomenon, has to happen when you have these stem cells around. Because once they're gone and you don't have enough of a stem cell population, if you give it that genetic hit, it won't develop a Wilms tumor, right? So these have a window of development. And by the time a child is 10 years of age, for example, you do see Wilms tumor in that age, but likely it's a different genetic etiology than the classical ones because that embryonal window is sort of closed. If we look at the genes involved in Wilms tumor, it looks just like the list I showed you for genes involved in renal development, not surprisingly, right? So you have WT1, Wnt beta-catenin, the Beckwith-Wiedemann locus, and then microRNA processing genes, histone modifiers, epigenetic modifiers, again, things involved in this primitive stem cell phenotype. As far as cancer predisposition in Wilms, we classically think of things like WT1 alterations, so like Wagar and Denny's-Drash, as being high risk. Medium risk, something like Beckwith-Wiedemann, and low risk, we have things like PROS, which I mentioned. So why then do we see Wilms tumor in PROS? PI3 kinase signaling is important in renal development. I'm not going to show a bunch of literature on this, but there isn't a lot of literature. There are some mouse models where they overexpress it, and lo and behold, when combined with other genetic insults, you can form Wilms tumors. Certainly known that there is increased PI3 kinase AKT signaling during this process of renal development, so it is important, but how it relates to the pathogenesis of Wilms tumor isn't really understood. So if it is important, then why don't we see Wilms tumor more commonly in PROS patients, right? If it is essential for development and it can cause that, why doesn't it cause it more? We do know that Wilms tumor patients have increased nephrogenic rest, the sort of precursors to developing, PROS patients have increased nephrogenic rest. But maybe the PIK3CA alterations are not sufficient for developing Wilms tumor. Maybe they need a second hit. Or maybe it's just the timing of things. The second hit occurs at a slow rate. Or maybe it's the microenvironment of having the PI3 kinase alteration first and then another hit. It sets it up completely wrong for development of Wilms tumor. Or maybe it's just most individuals don't have mosaicism involving their kidneys. So again, I don't really have any answers to this. I think it's rather fascinating to think about why these patients are getting Wilms tumors. And again, I think a lot more experience from other groups can hopefully help shed some light on this. Just a picture again. Ice climbing is relatively popular there. I did it once a couple of winters ago. That was enough for me. But it is a lot of fun, actually. A little scary. All right, I want to present one more patient to just expand the phenotype a bit, tie in a bit of the mosaicism as well as the Wilms tumor story, and maybe give a little bit more information on that. So this was a child we saw from the upper Northwest Territories who sort of came to us in Calgary, 22 months old, who said that, does this patient have PROS? Had this large right-sided epidermal nevus that you can see here, as well as overgrowth of the lower extremity. So did look like a PROS-like phenotype. However, had this angulation deformity of the tibia and fibulas, which was not typical. We don't tend to see those kind of skeletal anomalies within PROS. Developed bilateral renal masses, thought to be Wilms tumor, was treated and then had nephrectomy. In the course of the workup, a biopsy was done of the epidermal nevus, and this child was found to have a known oncogenic KRAS mutation in a somatic level. So again, a somatic mosaic, but not a PIK3CA, but the RAS pathway. And there is a lot of interplay between RAS and PI3 kinase signaling. They both sort of have a common end goal and pathways for growth, angiogenesis. So it's not surprising that we would see a RAS pathway mutation leading to a phenotype that's like that. This was the Wilms tumor. So this was the nephrectomy specimen. You can't even see any normal kidney here. There's really nothing. This fleshy rind on the outside is the nephroblastoma. All of that blastema precursor. And then these encapsulated masses are the Wilms tumor. And so there were encapsulated Wilms tumor here, nephrogenic rest, proliferative or adenomatous rest throughout. Another thing you might notice that's a bit odd for a Wilms tumor are these big vessels sitting here in the middle of the kidney, which aren't normal. And this kidney was riddled with a vascular anomaly that actually looked a lot like a FAVA. It was really weird. And it's the first time I've seen a Wilms tumor that has a vascular anomaly like that within it. So we did molecular on this and found that in the Wilms tumor, looking at both the Wilms tumor, the nephrogenic rest, the vascular anomaly, all had this KRAS variant present virtually in every cell, right? Because the allele frequency is high. So clearly the mosaicism needed to be very high to cause a Wilms tumor. And on the other side, because the patient had masses in the left kidney as well, and they were just taken out as small masses, there was a rim of residual normal kidney, which had that KRAS variant allele frequency at only 3.5%, suggesting that in the Wilms and these proliferations, there was a much higher rate of mosaicism. So we thought, okay. So clearly, you need a very high level of mosaicism to develop Wilms tumor. Maybe that's why it doesn't happen in PROS. But in this individual, we also found in virtually all the cells in the Wilms tumors, but not in other compartments, an FBXW7 hotspot mutation that's been associated with rare familial causes of Wilms tumor. So it looks like a second hit is probably needed to develop Wilms tumor, and that may be why the frequency is lower. I've seen this in at least 2 other PROS patients as well, where second hits were required to develop Wilms tumor. All right, so this patient was put together with a series of 8 patients that was published with this mosaic KRAS phenotype, mimicking PROS like with epidermal nevi, asymmetric overgrowth. They don't have the massive lipomatous overgrowth that we see in CLOVES, and they have this weird skeletal anomaly, which was always thought to be a fibrous dysplasia-like proliferation within the bones, but I've actually seen 2 of them now, and they're actually vascular anomalies within the bones that are causing these deformities. And this is a cancer predisposition, so these patients develop Wilms tumor. Rhabdomyosarcoma has been described in a couple now, and even a germ cell tumor. So clearly this is also a cancer predisposition like PROS. All right, so in putting together cancer predisposition and what I talked about today, because I really want to hammer this point home, we started talking about the PTEN Hamartoma Tumor Syndrome, which has a very high incidence of cancer predisposition, particularly carcinomas in early adulthood onto adulthood, right? I think these mosaic RASopathies lie definitely under that, where we see Wilms, rhabdomyosarcoma, germ cell tumors, so primitive, more pediatric type tumors, but at a higher rate than what we'd see in someone who has PROS, where we just tend to see Wilms tumor, at least for now. There are case reports of individual other tumors in patients. Whereas something like FAVA or KTS or even a benign lymphatic malformation, we're not going to see development of an aggressive malignancy. Interestingly, though, all of these conditions I've talked about have overgrowth and vascular anomalies, but none of them develop liposarcomas or angiosarcomas or any of that. So it's something about the pathways involved that are very different from the genetic etiology of those soft tissue sarcomas where they don't have an increased predisposition. So why is it that they're different? Why is it that something like PROS and PIK3CA are so different? I'll go back to my simplistic molecular diagrams here, which help me think about this. So if we think about PA3 kinase here sitting at the base of a receptor, it basically all it does is add a phosphate and then further downstream activate AKT-mTOR signaling, right, leading to our growth, migration, angiogenesis, vasculogenesis. PTEN does the opposite, right? It removes that phosphate and puts the brakes on the pathway. So we have one that's adding the phosphate and one that's removing the phosphate. So you would think that because it's such a similar function that the phenotype should be quite similar, but they're quite different, as I've shown you today. Now certainly PROS is due to type 1 mosaicism, whereas PTEN is a type 2. We have greater overgrowth in PROS, less in PTEN. PROS is a Hamartoma syndrome. Now PTEN classically is a Hamartoma tumor syndrome. I always like to think of PTEN as a hyperplasia tumor syndrome more. And then we have differing degrees of cancer predisposition. Why? Could be that one is constitutional, one is mosaic. There's redundancy issues and things. My simplistic way of thinking about this when I like to explain this is that, so PIK3CA is the gas on the pathway, right? So when you have an activating mutation in PIK3CA, you're stepping on the gas on this pathway. You still have PTEN, so you have a brake. Right now, you can go on with your car engine stepping on the gas and the brake at the same time. It's not great for the engine for long term, but you can slow things down to some degree, right? If you have a one-hit PTEN mutation, you already have bad brakes. If you acquire a second hit, no matter how much you let off the gas, if you're in drive, you can't stop, right? You're screwed. And so that's, I think, part of the difference. The other thing is there's almost no known functional redundancy for PTEN. So once you lose that brake, another molecule can't step up and provide that brake, where there are other things that can provide acceleration of this pathway. So I think there's many different factors going involved in that. Just a couple more pictures as I'm concluding. So in the fall, the hiking season is really fantastic up in Alberta, and these are the deciduous larch trees, which are a deciduous conifer. You have larches in the Cascades, right? Yeah. Siberian larch? I think there's a different species, yeah. Up in Alberta, the Siberian larches, and yeah, it just gets spectacular early on with this chartreuse color, then they turn bright yellow, and then get to this golden color usually just as the snow is starting to fall. Again, you've got to get to the hiking trails at like 5 a.m. if you want to get on the more popular ones, but many people have larches in their backyard, so it's not like, you need to hike to see them. All right. I'm almost done here. I want to wrap up that concept one other way, just because sometimes I think it hits home describing it in different ways. And my apologies to the neuropathologists and people who study these pathways. And this just ties into treatment at the end. So let's say we have a normal embryo here, goes through development, quote unquote, with no mutations, right? And then somewhere during life you acquire a somatic hit like a P53 or IDH and you get a glioma. You then give that glioma one of these PIK3CA hotspot activating mutations, and you can develop a lethal glioblastoma. You take that same PIK3CA alteration, you give it early in a low-level mosaic in the embryo, and you get these vascular anomalies and overgrowths, right? However, you don't develop brain tumors, even though you have overgrowth of the brain. So it's due to timing and the microenvironment at which these things are occurring, and likely the dosage also which these are happening. Now, back in 2012, when we first were finding these PIK3CA mutations, we went to Denise Adams, who's a big pediatric oncologist in vascular anomalies in our group in Boston, and said, look, we found this. Is there anything that can be done therapeutically for these patients? But luckily, because of all of these adult malignancies associated with PIK3CA, there were drugs out there, right? Like sirolimus, which is used as an mTOR blocker. And so therapeutic trials were started with treating these patients with these drugs. Again, I don't take any credit for this. This is all the oncologists and all the other people in the field doing this. And was started to be used more widespread for vascular anomalies with some degree of effect depending on the degree and condition. Now, nowadays, we have much more targeted therapies, again, from those adult malignancies associated with PIK3CA, things like alpelisib, which is a direct PIK3CA inhibitor, or something like trametinib, which is a MEK inhibitor, right, for the RAS signaling pathways, and other targeted inhibition, even GNAC inhibitors, which haven't been used yet in vascular anomalies, although I think they need to be for certain ones. And so now, more than likely, a combination of therapies can be used to treat these patients. I'm just going to show one slide from the French group's initial publication. This is out of the group in Paris on the use of alpelisib, the PIK3CA targeted inhibitor in PROS. And this was kind of their index patient treated for a year and a half. And I think you can see by all of these images there was a massive reduction in the overgrowth, the vascular anomalies, the epidermal nevi, all sort of melted down to a much more manageable state on a targeted inhibitor. But more importantly, this patient obviously was in severe cardiac and renal failure, and with treatment was much more manageable. So I think these targeted inhibitors are certainly the way treatments for vascular anomalies are going, to be able to just shut down those few bad actors. But more than likely for some of the more severe ones, because of all the crosstalk in these pathways, they're going to need dual level therapies and inhibitions. So that's sort of the future of where things are going. Not at all my area of expertise, so I'll leave that to others to talk about. And with that, I'd like to just stop and recognize all the hard work of the vascular anomaly centers that I've been involved in. So the group in Boston, where I trained and worked, and then in Calgary, and now in Utah. And then those members of my lab over the years who were instrumental in sort of doing some of this work I presented. So with that, I'd like to just stop and answer a few questions if there are any. So there's a question here from, hi Whitney, about why do you think cancer predisposition syndromes caused by a loss of a tumor suppressor gene, such as DICER1, P53, do not lead to vascular malformations in the same way that PTEN does. Yeah, I mean, that's a great question, right? Again, I think that's leading to the fact of why is it that PTEN and PIK3CA, well, you're talking about P53 and DICER1. Yeah, and I think that may just be due to the underlying pathways that those are involved in probably aren't as important to vasculogenesis as something like PIK3CA and RAS signaling pathways. And maybe that's why they're not leading to vascular malformations, in the same way that something like PTEN does. Although it's, again, interesting, but that's just sort of, a bit of conjecture. And then two, how does that differ from IDH mutated cancers from Ollier and Mafucci, which lead to okay, let's see. Oh, you mean, I think by that she probably is asking about, so the difference in predispositions with vascular anomalies and cancer predispositions like we see in IDH1. So for example, we were discussing this morning at pediatric slide rounds with something like spindle cell hemangioma, which is associated with IDH mutations and also can be as part of a syndromic association with Mafucci syndrome, with enchondromatosis, and that does carry a risk in the cartilage component at least of developing secondary chondrosarcomas, although the vascular components within that usually don't develop into more aggressive lesions than that. Yes. Okay. Yeah. Yes.
So thank you, Kyle. It was a wonderful talk. I mean, so clearly, I mean, to get to your point about cancer predisposition, there are the clinical and basic science questions here. I mean, one is there is varying degree. I don't know what you. I mean, there is some debate about who needs, because the thing is, if you find a PIK3CA mutation, there is a burden about do we need to survey them for Wilms tumor, and there are forms of 11p, loss of heterozygous where the risk is even as high as 40%.
Right, exactly.
Those need to be surveyed, but 3 to 5% is.
Yes.
So we have debated this. Is it is a burden to make someone come into the hospital and have an ultrasound every 3 or 4 months. And so I guess I wanted to speak to that. And then also, could you go into a little more about the statement that you made, I think, in this case where the Wilms tumor actually developed on the contralateral side to where the overgrowth was? And I thought, were you trying to suggest that there was non-cell autonomous signaling going on as well, or just that although the asymmetric overgrowth was on the right side or wherever it was, there still was PIK3CA mutation on the left side?
Yes. Yeah, no, that's what I was meaning. I think there was still PIK3CA on the other side, maybe at a lower degree of mosaicism, and that's why. But it's fascinating. Why did that side? So in that one individual, there was not on the contralateral side from the Wilms tumor, there were three independent Wilms tumors developing, but not that rind of nephroblastoma and all of that precursor stuff. So I think the mosaicism was quite different between the two kidneys in that individual, maybe leading to one to be much more severely affected and another to be less.
And on the side with the resected kidney, where
Yes.
you showed 3
Yes.
tumors, all 3 of those had the same PIK3CA and also the FBX?
2 of the 3 had the FBX. The other one we couldn't detect the FBX, and I'm not quite sure why. That one didn't have the best encapsulation either, and there's a, as pathologists, the distinction between a hyperplastic nephrogenic rest and a Wilms tumor really relies down to whether it's encapsulated or not, and so one could argue that maybe that's just the reason why there, but yes.
There is some thought, at least from measuring post-zygotic mutations in normal, non-cancerous tissues. Like, an example is notch mutations in the esophagus.
Yes.
There is some thought that early onset, sort of what we think of as activating mutations, can protect against cancer. So I'm wondering also if you thought about that with,
No, totally.
It's still even 3%, it's still lower than you would expect, right?
It's still lower than you would expect, and why is it that one would think if you have hyperplasia of your tissues, I don't care what it is, right? If you have an overgrowth of something, you clearly have some type of disordered proliferation going on, even if it's at a very low level. So one would think by adulthood, you should have some increased risk. But I think, yeah, it might actually be that it's more protective than anything else, because having that first sort of completely changes the genetic microenvironment there. And then if you acquire one of those hits, something happens. Maybe if you acquire a hit in one of those cells that already has a PIK3CA mutation, it's lethal right away and it can't develop, right? Because you're adding a mutation into an already, I have another slide that I show of this where I actually have an oncogenic seed that you plant into healthy soil, right? And it'll grow into a malformation. If you plant it into some with a few mutations, it might grow into a tumor. If you plant it into toxic soil, it's going to die, right? That seed's not going to grow. So probably it's something like that. If you acquire one of those other types of mutations in the background of one of these PIK3CAs, it might just be lethal. Yeah, and getting back to the screening, I think that's a great point. I mean, I think, and I don't know what you're doing here, but you know, if you have a child with PROS, and you do a screening ultrasound, and both kidneys are completely normally developed, no cysts, look completely normal, do you still follow them on the overgrowth protocol, you know, until they're 7 or 8 years old, or do you stop? You know, and I think that's a good question to really try and know, you know, because is it really worth it with that level?
Here locally, we've taken a sort of shared decision making.
Okay.
We're not rigid about it has to be every 3 months.
Yes.
So we kind of. And we've had a patient with Wilms, and we've also had a PIK3CA-related patient who had a lymphoma as well, and that's been. But
I think in Calgary we kind of did the same, and many of those kids, because we were sort of getting them regionally throughout Western Canada, and they were not always screened regularly. Especially now, the couple that had abnormal looking kidneys, yes, sure, then they were followed quite closely, yes.
Did any of your KRAS patients have CPAMs?
Good question. You know, you would think so, right? But none of those patients in the initial series had CPAMs, given that association again. It might just be due to, again, the mosaicism just doesn't involve the developing lungs. And if it's to that level, it might mean that it's across too many germ cell layers and it's embryonic lethal, right? Maybe some of the more, you know, neonatal fatal cases might have more lung abnormalities and other things, but yes. But no, it's a good thought. Okay. I think with that, we probably should stop, right? Because we're on. Yes, we need to get some.